Okay, so good evening, students. We are revising the chapter that is sexual reproduction in flowering plants. Okay, so we are revising sexual reproduction in flowering plants. Flowering plants means what? We are talking about the angiosperms. Okay, so in our last class, we have revised up to the pollen allergy and all. So we have discussed microsporogenesis, then the floral structure, then the gynoecium, androecium. So we have discussed up to that. So today we'll revise from NPC system of pollen classification. Okay. So this is a very important topic, NPC system of pollen classification. So what is NPC system? So N stands for nuclear, P stands for pollen, and C stands for cytoplasmic. That is called NPC. Nuclear, pollen, and cytoplasmic system is a widely used classification system for pollen grain based on their morphological characteristic and developmental aspects. So basically, the pollen grains are classified. So the most widely used classification system is this NPC system of classification. Okay, and who gave this? Who developed this classification system? Dr. K. Mehta. In which year? 1950s. Okay, during 1950s, Dr. K. Mehta developed NPC system of pollen classification. And this classification is very much important in identifying and categorizing pollen grains, and it facilitates studies in palynology. So yesterday we have discussed what is palynology. That is the study of pollen grain and other microspores. And it is very much helpful in botany, ecology, archaeology, then forensic science. So this NPC system is very much important. So let's revise what we have discussed earlier. So what are the components of NPC system of classification? Number one is the nuclear component. Okay, that is the N, the nuclear component. So in nuclear component, there is microsporogenesis, means the process of pollen formation, which begins with the MMC cells, and it will then undergo meiosis to produce haploid microspore. Then the nuclear feature of the pollen grains are determined by this process. Next is the ploidy level. So basically, pollen grains are haploid containing a single set of chromosomes. And understanding the nuclear characteristics, it will help the genetic makeup of the pollen. Next is nuclear arrangement. How the arrangement and the number of nuclei, like the generative nucleus and vegetative nucleus within the pollen grain, they can be vary. Okay, so this is the nuclear features in the NPC. Next is the pollen. Number two, P. Okay, NP. So what is pollen? So this pollen features refers to the external characteristic of the pollen grain. Okay, the external characteristic which are critical for identification and classification. So what are those external characteristics? The A is the exine structure, means the exine is, we have discussed that exine is a very tough outer layer made up of sporopollenin, okay? And it can be vary in thickness and ornamentation and composition related to species, okay? So a surface or ornamentation, in some you may find smooth, in some you may find spiny, in some you might find reticulate. So it will help in identification of the species. Next is the type of aperture. So we have discussed about monosulcate, trisulcate, polysulcate, means how many aperture are present within the pollen grain. So that is a type of aperture. Again, aperture plays a vital role in the classification. Next is shape and size, means the morphology of the pollen grains that can, from spherical, elongated, irregular shape, and size also varies. Next is the functionality, means the structure of the exine and aperture affects the pollen grain's ability to withstand environmental condition and interact with pollinators. So these all are the features of the pollen. That is the outer external characteristic. And the number third is the cytoplasmic, that is the C feature, NPC, nuclear pollen cytoplasmic. So C cytoplasmic features focus on the internal cellular components of the pollen grains. Okay, what C features do? It focus on the internal cellular components of the pollen grains. So cell contains, the cytoplasm typically contain two main cells. Okay, there are two main cells. One is generative cell and one is the tube cell. Okay, one is the generative cell. So we have discussed that the generative cell later on divides into two sperm cell which undergo double fertilization, and the tube cell, it will develop into pollen tube during the germination. Then the developmental stage, means the cytoplasmic structure, it will indicate the stage of development of the pollen grain. Okay, so it helps in researches means in the pollen viability, and how the pollen is ready for the fertilization process. So it comes under the developmental stage. Next is the metabolic activity. So the presence and abundance of organelles such as mitochondria. We know that mitochondria is the powerhouse of energy. Then plastids. So this provides an insight. This gives us an idea about the metabolic status of the pollen grain. So this cytoplasmic C features focus on the internal cellular components. That is what we call as NPC, nuclear pollen cytoplasmic system of classification. Based on these features, pollen grains are classified. So what are the significance of the NPC classification? So the significances are, number one, very important, is the taxonomic identification. Definitely, the system's main motive is to identify the pollen grains. So it helps in the taxonomic identification. Next is ecological studies. So you can, when you will understand the pollen characteristic, it helps ecologists to study the plant pollinator interaction and the dynamics of the ecosystem. Okay, and if there is some sudden change in environmental factors also, this all things are comes under ecological studies. Next is paleoecology. Ecology. So in paleoecology, the NPC classification assists in reconstructing past environments and climates by analyzing fossil pollen grains. So I have already told that pollen grains, they are also they preserved as fossils. Why? Because sporopollenin is present and it is very much durable. Okay, it easily didn't get degraded. So by studying paleoecology, one can reconstruct how past environments, our past environment is how, and the climate has changed with time. Next is the very important is the forensic palynology, of course. So the NPC can help in forensic sciences for criminal investigation and all, where the pollens give a link to the suspect, and also it helps. There are many cases where by studying pollen grains only one can find the criminal. Okay. So that is what NPC system of classification. So what is pollen viability? Okay, what is pollen viability? So pollen viability is very much important for the pollen because it refers to the ability of pollen grains to germinate and successfully form a pollen tube that delivers sperm cell to the ovule for fertilization. So if the pollen is not viable, then there is less chances of germination of the pollen tube and the failure of the fertilization. So pollen viability is a very much important factor in sexual reproduction of flowering plants. Okay, and viable pollens, it ensures that the male gametes are functional and capable of fertilizing the egg cell. Look, there are many pollen grains, but it is not mandatory that all pollen grains will be viable. Okay, so it is very important for the fertilization that pollen must be viable, means it must have the capacity to fertilize the egg cell. So what are the factors that influence the pollen viability? What are the factors? So number one is the age of the pollen grain. Okay, so generally the pollen grains have some limited lifespan. Okay, and the viability will decrease over time after they are set from the anther. Okay, so viability will be decreased with time. Okay, so it is very much it is inversely proportional. Okay, so it is viability will decrease if there is lot of time the pollen has taken from the set from the anther. So that is the one. And fresh release pollen is usually more viable compared to older pollen. Okay, next is the moisture. So pollen grains require certain moisture level to remain viable, and if there is excessive dryness can lead to desiccation, while too much moisture can also promote fungal growth. Look, it must be in optimal condition, okay, in minimum condition. We don't need too much dry pollen because it might lead to desiccation and degradation, while too much moisture can also promote fungal growth, okay. It will give an environment to the fungi. So moisture is an important factor for the pollen viability. Next is the temperature, okay. So pollen viability is affected by temperature. So high temperature can reduce viability by causing damage to cellular structure, while cold temperature may prolong viability up to a point. So basically optimal temperature is required. Still cold temperature is a bit, up to some point it is acceptable, but very much high temperature can cause damage to the cellular structure. Next is storage condition. So proper storage can maintain pollen viability for longer periods. Freezing or drying pollen can help preserve its viability. Next is nutritional factor. So the nutritional status of the plant influences pollen quality. So pollen produced under optimal growth conditions tends to be more viable. Now, how you will know that the pollen is viable? What are the tests to know the viability of the pollen grain? So here there are some common methods. Number one is the germination test. Okay. So pollen grains are placed on a suitable growth medium such as agar or nutrient agar and incubated under controlled condition. The percentage of pollen grains that germinate and form pollen tube is calculated. Okay, so the procedure is you will collect some fresh pollen grain, then you will prepare an agar medium with nutrients. Then you will place the pollen on the medium and incubate at optimal temperature and humidity. Then after a specific time, usually 12 to 24 hours, observe and count the number of germinated pollen grains. And the advantage is that it gives a direct measure of functional viability. This is most important, the staining test. Okay, so there are some stains used to know the pollen viability. So number one, common stains are fluorescent diacetate, that is FDA. So viable pollen grains fluorescence under UV light. When you will give this dye in the pollen grain, so the pollen grains which are viable, which are capable for fertilization, it will give a fluorescent under the UV light. Next is iodine staining, means stars grain with viable pollens can be stained, indicating metabolic activity. When you will give this iodine staining, what will happen in the pollen grains? The stars grain will be stained. It will take the color of the stain, and it will indicate that there is a metabolic activity going on the pollen grain. So procedure is this: mix the pollen grain with staining solution, then incubate for a short period of time and observe under a microscope. Okay, and see percentage of stained viable grains. So it is very quick and easy to perform and can assess a large number of samples. Next is our tetrazolium chloride test. So tetrazolium salts are reduced by viable cells, resulting in a color change. Viable pollen grains will turn red or pink due to metabolic activity. So what will happen? This tetrazolium salt, so this reduce the viable cells. So it will result in a color change. Okay, the pollen grains which are viable, it will turn red or pink due to metabolic activity. Next is pollen tube growth assessment. So this method assesses the growth of the pollen tube in real time, which is direct indication of pollen viability. So basically, the pollen grains will germinate, and if there is pollen tube, germination, there is growth of germination, then then the pollen is viable. Then feasibility indicators. So some plants release chemical signals while pollen is viable. This can be measured to determine viable indirectly. So it is an indirect method where some plants release some chemical signals. So change in specific metabolics or hormones related to pollen viability can be assessed using biochemical methods. So what are the examples of pollen viability? Tomato. Okay, tomato. So feasibility period is tomato pollen remains viable for about 5 to 7 days after dehiscence. So under optimal storage conditions, cool and dry, viability can be extended for several days. So in breeding programs, it has some practical implications. So it helps to determine the best time for pollination and hybridization efforts. Next is corn, our G-maize. So its viability is from 24 to 48 hours after it shed from the anther. And it is vine pollinated, and the timing of pollen release is crucial for successful fertilization. Next is the orchids. Okay. So the viable period is viable for several days to weeks. Okay. And it depends on species. So these are the thing about the pollen viability, which I have already discussed with you in the class. So I'm just revising the thing. Okay. So next topic is the sporopollenin, which is very much important. Sporopollenin. So what is sporopollenin? So it is a biopolymer that forms the outer wall exine of pollen grains and the outer layer of spores in seedless plants. Okay. This sporopollenin is a biopolymer, and it forms the outer wall of the exine of pollen grains and outer layer of spores in seedless plant. And it is the most chemically resistant organic material found in nature. Okay, so sporopollenin is the most chemically resistant organic material found in nature, and it provides essential protective function for pollen and spores during their transport and germination. So what is the composition? So sporopollenin is primarily composed of long chain fatty acids, phenolic compounds, and various other macromolecules, and its precise structure can vary significantly among different species. So sporopollenin is made up of what? Long chain fatty acids, phenolic compounds, and various other macromolecules. So insolubility, it is highly insoluble in most organic solvents and resistant to degradation by acids, alkalis, and enzymatic activities. This resistance is attributed to its complex and cross-linked structure. So basically, this is highly insoluble in organic solvents and resistant to degradation by acids, alkalis, and enzymatic activities. That's why they are resistant. That's why they are more durable and long-lasting. So what are the functions of this? It provides protection. Protection from what? Protection from desiccation, protection from ultraviolet radiation, protection from microbial attack. Okay. Then the facilitation of pollination. So sporopollenin can influence the adherence of pollen grains to pollinators, enhancing successful fertilization. Next is preservation of the fossils. It is the most important point. So due to the durability of the sporopollenin, it allows the pollen and spores to be preserved in sediments for thousands to millions of years. This property is invaluable for paleobotanical studies, and it provides insights into historical climates and vegetation. As due to the durable characteristic of sporopollenin, it is preserved as fossils. Okay, it is preserved as fossils. And biological significance. So the presence of sporopollen in the exine of pollen grain contributes to the evolutionary success of seed plants, enabling them to reproduce effectively in diverse environments. So this is the conclusion. So we have discussed almost everything in the male part, starting from microsporogenesis to the structure of androecium, to the palynology, structure of pollen, then the classification of pollen and species system, then the sporopollenin, pollen energy. So everything we have discussed, and all things are very much crucial in the field of agriculture, horticulture, and conservation biology, where pollination and plant reproduction plays a pivotal areas. So it is very much important. Okay, so now we will start, we'll discuss, we will revise the topic megasporogenesis. Okay, we are done with microsporogenesis. Now we'll discuss megasporogenesis. So what is megasporogenesis? So it is the process in which the megaspore, that is the female spores, are produced in plants, especially in ovules of seed plants. Micro means microsporogenesis, where we will get microspores. That is the male spores. But megaspores, mega means big. Means you look one thing, remember one thing, that a megasporogenesis, mega is used because the spores are larger than the microspores. That's why it is termed as megasporogenesis. So here it is the process where you will get the female spores. So let's have the overview of megasporogenesis. So there are some sequence of events which will lead to the formation of megaspore, which are the haploid cells that will develop into the female gametophyte. And in most of the plants, this process involves meiosis and occurs within the ovule, where it is precursor to the seed. So what are the steps? Number one is initiation of megasporogenesis. So megasporogenesis begins with the differentiation of specialized cell called the megaspore mother cell, same like the micro one. So MMC, which is typically found within the ovule. So the ovule develops within the ovary of the flower. The MMC, so we have known that the MMC is a diploid cell, and it contains two sets of chromosome, one from the mother and one from the father. So first there will be initiation of the megaspore mother cell. So what are the key points? So MMC originates from the cells of the nucellus, which is the tissue surrounding the embryo sac in the ovule. So it is a very important question, from where MMC's megaspore mother cell, MMC cell originates. So it originates from the cells of the nucellus, which is the tissue surrounding the embryo sac in the ovule. It is originates from the nucellus. So the formation of MMC is initiated by the differentiation of the diploid cells in the ovule, typically influenced by hormonal cues in the plant. So then after that, there will be meiosis, okay? Same like the microspore mother cell. So here will be meiosis, okay? So which results in the production of 4 haploid cell. So same thing, okay, like microsporogenesis, here also there will be meiosis. So in the meiosis one, you will first get 2 daughter nuclei, and at the end of meiosis 2, you will get 4 haploid daughter cells. What is the difference between microspore and megaspore? So the main difference is that in microsporogenesis, the all 4 haploid daughter cell, all 4 haploid cells are functional, but in megasporogenesis, in the megaspores, here out of the 4, all 3 will degenerate, but only 1 will be functional. Means only 1 will survive and rest of the 3 will degenerate. Okay, only 1 will survive. This is the key difference. So development of megaspore. So out of the 4 haploid megaspore produced during meiosis, typically only 1 survives and develops into female gametophyte, while the others will degenerate. But in our earlier microsporogenesis, what we have seen, all the 4 are functional. Okay, but here only 1 is functional and it will develop into female gametophyte, while the rest will degenerate. Okay, so the key points is that the surviving megaspore is usually the one closest to the micropyle, the opening of the ovule where fertilization will occur. So generally, which daughter cell will survive? The daughter megaspore, which is usually the closest to the micropyle end. So basically, there are 2 ends in the gametophyte. One is chalazal end, one is the micropyle end. So the cell which the megaspore which is closest to the micropyle, it has more surviving chances. What is micropyle end? It is the opening of the ovule where fertilization will occur. And the other 3 megaspore which are further from the micropyle are discarded or degenerated. Then the surviving megaspore will undergo mitotic divisions to form the female gametophyte embryo sac. So formation of the female gametophyte. How female gametophyte is formed. Listen very much carefully. Okay, understand this concept because it is very much important. So after the selection of the functional megaspore, this cell undergo a series of mitotic division. Okay, now there will be mitotic division, means repetitive division, but no cytokinesis occurs during the initial divisions. Means there will be no cytokinesis in the initial, means in the first divisions. So as a result, the cell become multinucleate and the embryo sac or the female gametophyte begins to form. So basically, how many mitotic division will be there? There will be 3 mitotic division to form a female gametophyte. The functional megaspore undergo 3 rounds of mitotic with mitosis without cytokinesis, and this results in the formation of a multinucleate structure known as megaspore mother cell. So after the division, cellularization occurs, meaning that the cytoplasm partition around its nucleus, resulting in a mature embryo sac, which typically consists of sorry, which typically consists of 7 cells and 8 nuclei in case of angiosperm. So in the case of a typical angiosperm, the female gametophyte will consist of 7 cells and 8 nuclei. So I will describe the structure of the female gametophyte, that is the embryo sac. 7 cells, 8 nuclei. Okay. So in most angiosperm, the mature female gametophyte is called the embryo sac, and it is consist of 1 egg cell. So this is the cell that will fuse with the male gamete sperm during fertilization. So this egg cell will fertilize with the female male gamete, that is the sperm, during the fertilization. Next is there will be 2 synergid cells. Means these cells are positioned adjacent to the egg cell and helped in guiding the pollen tube towards the egg for fertilization. So egg cell will be in between, and surrounding the egg cell there will be 2 adjacent synergid cells. So it will guide the pollen tube towards the egg for fertilization. Next is there will be 1 central cell. So this contains 2 polar nuclei and will eventually fuse with 1 sperm cell during double fertilization to form the triploid endosperm. So this 1 central cell, it has 2 polar nuclei. It has 2 nucleus, and it will fuse with 1 sperm cell during double fertilization to form the triploid endosperm. So egg cell will fertilize with one male gamete, and the central cell will fuse with the next second male gamete. That's why it is called as double fertilization. And there will be three antipodal cells, okay, which will be in the chalazal end. So these cells are located at the opposite end of the embryo sac, away from micropyle, and their exact role is still debated, but they may degenerate in some species. You can see this is the diagram. It will be more clear. You can see in the bottom position, the central egg cell is there, okay, and the egg cell is surrounded by the two synergids, okay, two synergid cell. Then in the between, there is the polar nuclei, the central cell, and in the above, there will be the antipodals, okay. In the chalazal else, there will be antipodal. So count the cell, 3, 3 plus 3 equal to 6, and in between, there is a 1, okay, 7. That's why it is called as 7 cell structure. And count the nucleus. Above there are 3 nucleus. In below there are 3 nucleus. 3 plus 3 6, and in the middle one, in the central cell, there are 2 nucleus. That's why 6 plus 2 8. So 7-celled 8 nucleus structure. So a typical embryo sac contains 7-celled 8 nucleus structure. Let's discuss the summary of megasporogenesis. So there will be differentiation of megaspore mother cell, MMC. Then there will be meiosis, okay, give rise to 4 haploid megaspore. Then 1 will functional, 4 will degenerate. Okay, the one which is closest to the micropyle end will survive. Then there will development of female gametophyte. There will be 3 mitotic divisions, okay, and it will form a multinucleate structure. And there will be formation of the multinucleate structure, where there will be 1 egg, 2 synergid cells, 1 central cell, and with 2 polar nuclei and 3 antipodals. 7 cells, 8 nucleate structure. So this is what about the megasporogenesis. So significance of megasporogenesis is genetic diversity. So the process of meiosis ensures that the resulting megaspores and eventually the embryo sac contains a unique genetic composition. And this is important for maintaining genetic diversity within the plant population. Okay, it is important due to the genetic diversity. Then foundation for fertilization. So the egg cell produced through megasporogenesis is essential for fertilization, because if there is no egg cell, there will be no fertilization. Now comparison with microsporogenesis. Okay, so while megasporogenesis involved the formation of the female gametophyte, microsporogenesis is the process by which male gametophyte pollen are produced in anther. So both process involves meiosis, but the key difference is that in megasporogenesis, only 1 out of the 4 produced spores survives, while in micro, all 4 microspores usually survive and give rise to pollen growths. This is the key difference. Okay. So this is the conclusion, that is very much highly regulated and essential process in the reproductive cycle of seed plants, and it ensures the formation of the female gametophyte, which is crucial for fertilization and seed development. And understanding the megasporogenesis not only help us understand plant reproduction, but also provides insight into the mechanism of genetic variation and evolutionary process in plants. So I have already in megasporogenesis, I have already teach you all thing about the megagametogenesis also. So means basically what is the term is different, that in megasporogenesis what happened up to the formation of the megaspores, and megagametogenesis is the process, the formation of the development of the female gametophyte, means the from the megaspore to the gametophyte, means the 3 mitotic division stage, these all are come under the megagametogenesis. So these are the things that okay, fine. So selection of the functional megaspore, that only 1 will survive. Okay, the rest 3 will degenerate. Then there will be first mitotic division, formation of a multinucleate structure. Okay, it will form 2 nuclei. Then there will be second mitotic division. It will form 4 nuclei. Then there will be third mitotic division. It will form 8 nuclei. That's why it is known as 8 nucleate 7-cell structure. This is the thing. And cellulation and formation of the mature embryo sac. So after the third mitosis, the embryo sac undergoes cellularization, meaning the cytoplasm will divide and each nucleus gets surrounded by a membrane to form individual cell. Then you will get the individual cell, that is the egg cell, the 2 synergid, the central cell. Okay, and the 3 antipodals. You can see the diagram. These are the thing. So number one, basically there are special type of embryo sex also, but I have discussed you with the everything is same. I have discussed about the polygonum type, where you can see that there is from 1 to 2 to 4, 4 to 8, then 3 in 8, 3 will go above, 3 will go bottom, and 2 will remain in the middle. That is polygonum type. But there are many more types of embryo sex present in the gynecium. So key difference between megasporogenesis and megagametogenesis is what? Megasporogenesis is the phenomenon where the formation of megaspore from the megaspore mother cell through meiosis. And this process is precursor to megagametogenesis. And megagametogenesis is the development of the female gametophyte. This is the embryo sac from surviving functional megaspore, and it involves 3 mitotic divisions, and the cellular differentiation requires for fertilization. This is the difference. So sorry, this is not he, this should be da. Typing mistake. So the embryosic is the female gametophyte in angiosperm flowering plants, and it plays a crucial role in plants' reproductive process, and it develops within the ovule and is involved in fertilization. Okay. And the embryosic consists of 7 cell, 8 nuclei in plants, so some species may have variations. And the cells are arranged in a specific way to facilitate fertilization by the male gamete, that is the pollen grain. So I have already discussed about the egg cell. So what a egg cell position, it is located in the micropyle end of the embryo sac, and it is the closest region to the entrance of the pollen tube. And its role is to fuse with the male gamete to form the zygote. Okay, and it is typically surrounded by the 2 synergid cell, which play a role in directing the pollen tube. Next is the synergid cell. Its position is adjacent next to the egg cell at the micropylar end of the embryo sac. And role is the synergids assist in guiding the pollen tube towards the egg cell during fertilization. Means they release some chemical signals to attract the pollen tube and help facilitate the fusion of male and female gametes. And details is that the synergids contain a special structure called the filiform apparatus. Why? Because it aids in the attraction of the pollen tube. And after fertilization, the synergid cells will degenerate. Next is the central cell. It is located in the middle of the embryo sac opposite the micropylar end and contains 2 polar nuclei. Okay, it in the central position. Its role is to fuse with the second male gamete to produce a triploid endosperm. Okay, why triploid? Because already central cell is diploid, then again it will fuse with a male gamete which is haploid, means 2 plus 1 is equal to 3. That's why the product is the triploid endosperm. So details is that the central cell is crucial for forming the endosperm, and endosperm is the tissue that will nourish the developing embryo in most plants. So the 2 polar nuclei fuse to form with a diploid nucleus when fuses with the sperm cell to form the triploid endosperm. Next is the antipodal cells. So these are located at the chalazal end, opposite end of the embryo sac. And role is it is thought that its role is to play a role in the nutrient exchange and may help in development of the embryo sac. Okay, but exact function is not fully understood and believed to generate after fertilization. So typically there are 3 antipodal cells in most plants, and they serve mainly a structural and supportive function. Embryosic development, monosporic polygonum type. So already these things I have discussed. This is what I have discussed about the megagametogenesis. So next is the fertilization. So pollen tube penetration. So the pollen tube grows down the style and enter the embryo sac through micropyle, where it will release the 2 sperm cells. Okay, I will discuss it in the pollen pistil interaction part. Don't worry, I will revise it again. So basically, in fertilization, what happened? The one male gamete will fuse with the female egg to form the zygote, diploid zygote, whereas the second male gamete will fuse with the central cell with 2 polar nuclei to give the product triploid endosperm. That is a trisome. That's why it is known as double fertilization. What are the products? A zygote, a diploid zygote, which will turn into embryo later on and eventually grow into a new plant, and endosperm, which nourishes the developing embryo and become a part of the seed. So What are the types of embryo sac development? So basically, it varies in different species, with the polygonum type being the most common. So most common is the polygonum type, which I have discussed with you right now. So other variation exists based on number of megaspore involved. So monosporic type, there is the polygonum type. One megaspore forms the embryo sac. Means only one megaspore will, out of the four, three will degenerate and only one megaspore will form the embryo sac. 7 cell, 8 nuclei. Next is the bisporic. So here two megaspores contribute to the formation of embryo sac, often seen in plants like allium, onion, and endymion. So basically in bisporic, what happened, out of the four haploid megaspore, two will participate and two will degenerate. Okay, and that two will participate, and it is seen in allium, onion, and endymion. So it is bisporic type of embryo sac. And this is the tetrasporic. So here all four megaspore will participate, means nothing will degenerate. Okay, in the formation of the embryo sac, leading to more complex nuclear division as seen in adducts, plumbago, and peperomia. Okay, so these are the types of embryo sac development: monosporic, bisporic, and tetrasporic. But monosporic is the most common, and polygonum type is the most common. 7 cells, 8 nuclei. So the ovary, now we'll discuss about ovary. So ovary is a critical part of female reproductive system in angiosperms, flowering plants, and it houses the ovules. So within the ovary, you will find the ovules, which will develop into seeds after fertilization. So ovules developed into seeds after fertilization. So the ovary is located at the base of the flower and is a key structure in the formation of fruit. So ovary is the located at the base of the flower, and it is the key structure for the formation of the fruit. So what is the structure of ovary? So it can be described in terms of external structure, internal structure, and specialized region. Okay, the structure of ovary can be described in terms of external, internal, and specialized. So what are external structure of the ovary? The ovary is usually a swollen basal part of the pistil, female reproductive organ, located beneath the stigma and style, and it is part of the flower that contains one or more ovules. Okay, so shape and size. So the ovary may be spherical, cylindrical, or pear shaped. It depends on the species. Okay, and the shape is often influenced by the number of ovules and number of carpels. And size of the ovary can be greatly depending on plant species. For example, in some species the ovary is very small. Example, in grasses, while in others it can be quite large in apples or cucumbers. The number of carpels. So Sembert structure. So the ovary is typically composed of one or more carpels, the female reproductive unit, and the number of carpels determine the number of ovary chambers or locules. Means the number of carpels will decide the number of chambers of the ovary. So there will be unicarpellary, means only a single carpel forms the ovary, and there is only one locule. Example is P. Then multicarpellary, means more than one carpel is fused to form a compound ovary and multiple locules are present. Example, tomato or cucumber. Next is syncarpous ovary. Multiple carpels are fused together to form a single ovary, that is tomato. Here, multiple carpels will fuse to form a single ovary. Next is apocarpous ovary. So carpels are separate and do not fuse, leading to several ovaries. Example, strawberry. So in strawberry, carpels are not fused, they are separate, and it leads to several ovaries. This is ovarian wall, pericarp. So the ovary wall is known as the pericarp. What is pericarp? The ovary wall is the tissue that surrounds the ovules and eventually develops into the fruit after fertilization. So pericarp is composed of three main layers: exocarp, mesocarp, endocarp. Exocarp is the outer layer, and it is the outer layer of the ovary, and that often develops into the fruit skin. So the fruit skin is the exocarp. Next is the mesocarp. That is the middle layer. So middle layer often fleshy and edible in many fruits. The flesh of peaches. Okay. So this is the edible. Next is the endocarp, the inner layer. So innermost layer which surrounds the ovule and may harden to form a pit or seed coat in some fruits. Okay. This is the inner layer. Now the internal structure of the ovary. So internally, the ovary contains one or more ovules which develop into seeds after fertilization. So internal structure include ovules. So ovules are the female gametes, which is the potential seeds. Okay, and it is located within the ovary, and each ovule is connected to the ovary wall via a stalk called the funiculus. Okay, ovule is connected to the ovary via a stalk known as funiculus. So what is the structure of an ovule? So it has integuments. So integuments are the protective layers that surround the ovule. And in most plants, there are two integuments, inner and outer, that will form the seed coat. Next is the micropyle. So it is a small opening in the ovule through which the pollen tube enters during the fertilization. Next is the nucellus. So nucellus is the central tissue, the central body of the ovule, which contains the megaspore mother cell, the precursor to the female gametophyte embryo sac. And next is the funiculus. It is a stalk that attaches the ovule to the ovary wall. You can see the angiosperm part. So this is the megasporocyte, nucellus, micropyle, and this is the funiculus, which attach the ovary to the ovule to the ovary. And these protective sheets are the integuments. Okay. Next, placenta and placental tissue. So placenta is very much important. It is the region of the ovary where the ovules are attached. So placenta is a region of ovary where ovules are attached. Okay, so there are some arrangement of placenta like parietal placentation where ovules are attached to the inner wall of the ovary. Then axial placentation where ovules are attached to a central column of the ovary, creating a multiple locules, that is the chambers. Next is precental placentation. Ovules are attached to a central column but without dividing the ovary into locules. Next is basal placentation. Ovules are attached at the base of the ovary. Okay, here the ovules are present at the base of the ovary. Then marginal placentation. Ovules are attached along the margin of a single carpel. That is common in P. So basically, these all things are like type of placentations. Based on this, you can also identify. Okay, what type of ovule is this? So you must remember the term. Next is ovarian chamber. This is the locule. So I have already discussed there are unicarpellary and multicarpellary. Were made up of one carpel or more than one carpel. Types of ovary based on position. So it is very much important. So there are types of ovary based on, we can classify the types of ovary based on position. Okay. So ovary. So number one is the superior ovary. So what is superior ovary? So ovary is positioned above the insertion of the other floral parts. That is petals, sepals, and stamens. Means ovary is positions above these parts. Okay. Example is lily and tomato. Here the fruit develops from the upper portion of the flower and the sepals, petals, and stamens are attached below the ovary. So the sepals, petals, and stamens will be attached below the ovary. That's why it is called as superior, above, superior ovary. Next is inferior ovary. This is vice versa of the superior one. Here ovary is positioned below the attachment of other floral parts. This means the sepal, petals, and stamens are attached above the ovary. Okay, so here ovary will be in the below part of the sepals, petals, and stamens. So example is apple and cucumber. So the fruit develops from the lower portion of the flower and the sepals, petals, and stamens are above the ovary. That's why it is known as inferior. Next is half inferior or half superior ovary. This is an intermediate position where the ovary is partially below the attachment of the floral parts. Means its position is in the, medium. That's why it is partially below the attachment of the floral parts. Example is peach in some spaces and pomegranate. This is development ovary into fruit. So after fertilization, the ovary develops into the fruit which contains the seed. Okay, ovary will develop into fruit and it contains the seed. The development of the ovary into fruit can occur in several ways. So simple fruit, so a single ovary can develop into a fruit such as peach or tomato. Then aggregate fruit, multiple ovaries from a single flower fuse to form a fruit such as strawberry or blackberry. Means multiple ovaries from a single flower will fuse to form a fruit. That is known as aggregate fruit. Next is multiple fruit. The ovaries from multiple flowers combine to form a single fruit. So here, what is the difference of aggregate and multiple fruit? In aggregate fruit, multiple ovaries from a single flower will fuse. Okay, but in multiple fruit, the ovaries from multiple flowers combine to form a single fruit. So example is pineapple or fig. Okay. So today we'll conclude till here our revision, because next part is there is some, again there is a different part. So today we'll conclude till here. Okay, development of the ovary into the fruit. So our revision is completed till here. So what we have discussed today, we have discussed about the megasporogenesis, megagametogenesis, then ovary, about ovary structure, about ovule structure, then types of ovary based on position. Okay, so we have discussed these things in today's class. Okay. Okay. Okay, student. Then in the next class, I will revise the remaining topics. Okay. Hope till now it is cleared.